Hello students. Today let us begin with the dihybrid test cross. In the previous class we have discussed the dihybrid cross. Fine. So dihybrid test cross. Here F1 we are going to cross with the recessive parent. F1 dihybrid heterozygous, which is heterozygous in condition, we are going to cross this with the recessive parent. So already I told you what do you mean by test cross, why it is done. So what is test cross? It is a method to know the genotype of unknown individual, and this we will do by crossing the unknown with the recessive parent. So in the dihybrid cross, the F1 is heterozygous, round and yellow. The cotyledons were having the yellow color, and recessive parent is wrinkled and green. So this we have to consider as parent. So let me write little this side. We write little this side. So what parent we have taken? Heterozygous F1 is crossed with the, so we can consider this as parent, or simply we can take the individual which is under test. Let me take like this. Now the gametes. So here I told you how to take the gametes. Now first let me take, you have to cross this with these two and this with these two. So capital R, capital Y, capital R, small r, capital Y, and small r, small y is crossed with the recessive parent. As the recessive, they are homozygous in condition and they will give only one type of gametes. So no need for us to write all the four gametes. Let us cross. So how many gametes are there? Those many rows and columns you have to draw. So here I am taking only one gamete, because all the progenies which we are getting are same. No need for us to write all those. Now let me take this as male and this as female. Let me draw 4 columns. Write all the 4 gametes. Already you have learned how to cross, and always you have to remember that the first which character we have considered, that character you have to write, and among that you have to write the dominant one first. So I wrote the genotype. Let us say here down all you will get same only, all 16 you will get like, here 4 you will get same, 4 you will get same, same like that only. Now let me write the phenotype. Round, yellow, round, green, wrinkled, yellow, wrinkled, green. So here all the type you will get 1. Round yellow you got, round green, wrinkled yellow and wrinkled green. All 4 you will get. Let me write the phenotypic ratio. Phenotypic ratio. So here you will get 1:1:1:1. That is 1 round yellow you will get. Round yellow 1 you will get, and round green you will get, wrinkled yellow you will get, and 1 wrinkled green you will get. Wrinkled green you will get 1. So all 25%. If you take all this as 100%, 25% different kinds of plants you will get. And when you take the genotype, when you take the genotypic ratio, that also you will get 1 is to 1 is to 1 is to 1. Fine. So here you will get genotypic and phenotypic ratio same. So when you will get all 4 types of plants in the progenies, then we have to understand that the individual which is under test, is heterozygous in condition. That is, it is heterozygous for the given condition. So here the individual under test is heterozygous in condition. Heterozygous in condition. Fine. So individual under test is heterozygous in condition. So what did I told now? Dihybrid test cross. What do you mean by test cross? It is a method to know the genotype of unknown individual. So as here it is, everything is clear and we know that F1 is heterozygous. So during Mendel time it was not known, so he gave us the result. Fine. So unknown, or let us take the F1 which is crossed with the recessive parent. When you cross the F1 with the recessive parent, you will get genotype and phenotype in equal ratio. That is 1 is to 1 is to 1 is to 1. So it is understood that the F1 individuals are heterozygous. Suppose if the individual which you have taken, let me take it as a second case. So if the individual which you have taken is homozygous for the given condition, then how you are going to get the result? So homozygous dominant. Let me take homozygous dominant. So now from the known, we will just see what kind of ratio we are going to get. So homozygous dominant, if we get, cross this with the recessive parent. So homozygous dominant here is capital R, capital Y, and cross with homozygous recessive. So here you are going to get only one type of gamete as they are homozygous. And progenies, how do you get the progenies? So all you will get same variety, that is all are tall. So all are tall. So that is when you draw the table, you will get same variety. The gametes are same. When you draw a table like drawing 4 rows and 4 columns, all 16 will be tall. You are not going to get all 16 will be of round and yellow shape. They will be having the round shape and yellow color. You will not get wrinkled and green one. That means, what is the phenotypic ratio here? Phenotypic ratio is 16 is to 0, and genotypic ratio is 16 is to 0. You are going to get only one variety. So when you will get like this, dominant character only, no recessive characters at all in the result, then the individual which we will take for the test is homozygous dominant. It is one case. Hope understood. Now let me go to the next one. Now let me take the third case. Homozygous recessive, let us take. Homozygous recessive crossed with a recessive parent. Now homozygous recessive is small r, small y, and recessive parent is okay. Gametes, what kind of gametes will get? Same. And what kind of progenies you are going to get? Capital R, capital R. So all will be wrinkled green. So in the third case, we have taken the homozygous recessive individual, which we consider that as a unknown. Suppose that if we take the wrinkled yellow color wrinkled yellow color seed giving plants, when we take and cross those flowers with the recessive parent, so we are going to get only the recessive characters, that is wrinkled green. So what kind of ratio you got? Phenotypic ratio. Phenotypic ratio will be, you are not going to get any dominant character. 0:16 you will get, and genotypic ratio also. Genotypic ratio also you are going to get 0:16. Then when you get this kind of ratio, what you have to understand, that the individual which is under test is homozygous for a given character, and that too it is having the recessive traits. It is having the recessive factors. It is giving recessive character, and recessive characters are expressed under homozygous condition. So these are the three different kinds of test ratio which we will get under three different cases. Like under heterozygous condition, when both the factors are heterozygous in condition, then we will get the ratio. Genotypic and phenotypic ratio as 1:1:1 we will get. And if it is homozygous for dominant character, then you will get 16:0, both the ratios. And when it is recessive for the given character, then you will get 0:16. Sometimes the ratio may be given and the individual which is under test can be asked in the CET. Hope it is clear. Now in the monohybrid test process, I did not told you that these two cases. So let me just say you. Hope it is clear. Now let me say you the monohybrid test cross ratios. Well, let me just say you the ratios of monohybrid test cross. See, under heterozygous condition, so heterozygous, if the unknown is heterozygous, let me take this side. If the individual which we have taken for the test is heterozygous, you will get phenotypic and genotypic ratios as 1:1. In equal ratios, you are going to get the tall and the dwarf. And if it is the first case. Second case, if it is homozygous dominant. If it is homozygous dominant, then what kind of ratio you will get? Phenotypic ratio. You will get 1:0 you will get. And genotypic ratio also, all the type will be same. You will be getting 1:0. No tall trait at all. Now third case, if it is homozygous recessive. If it is homozygous recessive, phenotypic ratio, and genotypic ratio. So 1:0, let me write 4, because here you will get 2:2. So here you will get 2:2. So by simplification we will write 1:1. Here also you can write 1:0 you can write. Even this also will do. So if you take, so we were discussing the third case, homozygous recessive. In the homozygous recessive phenotype, you will get all dwarf variety only. That is no tall variety. By simplifying, you can write 0:1 and genotypic ratio also same. You'll get all recessive character. So if the genotypic or phenotypic ratio will be given and asked you to write the phenotype of the genotypic condition of the individual, you can write whether it is homozygous, heterozygous for a given character. Hope it is clear. There is no need for us to cross this and say. Then also let me write the crosses. Capital T is crossed with the recessive parent. So all you are going to get the heterozygous tall. That is all the genotype and phenotypes will be same. And here you are going to cross the homozygous recessive and all. What kind you will get? You will get all the recessive parent. So these are the test cross ratio in monohybrid cross. Fine. Just you have to remember when you get the result like this, the genotype will be what I wrote here. When you get all 1:1 ratio, it is heterozygous. When you get 1:0, then it is homozygous dominant. When you get 0:1, then it is homozygous recessive. Now let us see the next one, that is incomplete dominance. Okay, now let us discuss the deviation from Mendels Law. So during his time, many of the plants and animals obeyed Mendels Law. That is when they crossed the organisms by considering one character. They got phenotypic ratio as 3:1 and genotypic ratio as 1:2:1. And in the dihybrid also they got the same as Mendel got, like 9:3:3:1, and genotypic ratio also he got same. The other scientists have got the same. But some of the organisms did not obey Mendel's laws of inheritance. The principles they did not obey. Among them, 4 we are discussing, which are given for the PU syllabus. So those phenomena were called as incomplete dominance, codominance, multiple alleles and pleiotropy. Let us discuss one by one. First, let me take the incomplete dominance. So, incomplete dominance. So, incomplete dominance is also called as partial dominance. It is also called as partial dominance, or it is also called as blended inheritance. Blended inheritance. And for the first time it was studied in detail by Karl Correns in Mirabilis Jalapa. In Mirabilis Jalapa, which is called as 4 o'clock plant. 4 o'clock plant. And in the NCERT, snapdragon flower is given as an example. Snapdragon, or you can say Antirrhina species. Antirrhina species. So Karl Correns, for the first time, when he crossed the flowers of the snapdragon, or you can take Mirabilis Jalapa. So here you will get 2 kinds of flowers, that is red and the white. So first what character he took? Flower color. Flower color. That is red flowered plants he took and white flowered plants he took. And he indicated the factors with the capital letters of the dominant one, capital R, small r. And he followed the Mendel's experimental procedure only. What kind of parent he selected? Homozygous parent he selected. That is red flowered plants, red flowered and white flowered. White flowered he selected. Let us write the genotype, gametes, capital R, small r. Now F1 progeny or F1 generation, let me write. F1 generation. In the F1 generation, all were heterozygous and they did not give either of the parental color. They were pink in color. All were pink. All are, let me write, pink. All plants were giving the pink flowers. And now for F2 generation, what he did next? He crossed F1 among themselves. Let us cross here. So gametes. As they are heterozygous, two kinds of gametes these plants will give. And F2 generation. Let me write the F2 generation. Let us draw the Punnett square just to avoid the confusion. One side male, one side female, let me take. Capital R, capital R, capital R, small r, capital R, small r and small r, small r. Let me write the phenotype. Here it will give red flowered plants, pink flowered plants, pink flowered plants and white flowered plants. So when you see the phenotypic ratio, when you see the phenotypic ratio, you'll get 1:2:1. That is red one plant, 25%, and pink you'll get 50%, and white again 25%. That is red 25%, pink 50% and white 25% he got, Corin's got. That is 1:1:1 ratio he got. Let me see the phenotypic ratio also. Now we were discussing about the incomplete dominance, and in the incomplete dominance, I told incomplete dominance it is a deviation from the Mendels Law where Corin's did not get the ratios as Mendel got. So what example he took? Snapdragon flowers. So here the red flowered plants were crossed with the white flowered plants. So and this is indicated by capital R and white is indicated by small r as usual. So when these two are crossed in the F1 generation, Corin's got all pink flowered plants. He did not get either of the parents, neither he got red nor he got the white. An intermediate character he got in F1 generation. So when he followed the next step, that is third step, selfing, self-pollination. When he selfed the F1 individuals among themselves to get the F2 generation. Here he got the flowers in the ratio of 1:2:1. That is 25% he got the red variety, 50% he got the pink variety and another 25% he got the white varieties. So the red and white which did not appear in the F1 generation he got in the F2 generation. So here when you see the phenotypic and genotypic ratio, phenotypic ratio also he got 1:2:1 and genotypic ratio also he got 1:2:1. So here both phenotypic and genotypic ratios are same. So thus it is the result or it deviated from the Mendel's regular laws. So this kind of blending of characters, when both the alleles are present together, the dominant gene is not it is not completely dominant over the recessive, or you can say it did not express itself completely, and neither the recessive got completely suppressed. So both of them got expressed together, the characters got mixed, and it expressed an intermediate color to their parent. So such a process is called as incomplete dominance. What do you mean by incomplete dominance? It is a 5-mark question asked in the annual exam. So you have to write all these. What you have to write? So incomplete dominance, by definition, it is a process where the dominant gene is incompletely dominant over the recessive gene, and it produces an intermediate character. It produces an intermediate character. So it did not indicate the parental type. Fine. So this is the definition. What example you have to take? Snapdragon. It is given in the NCERT. So let us write this only. And what character you have taken? The flower color. How many flower colors are there in the anti-random species? 2. Red and the white. Fine. So when you cross in F2 generation, what kind of individuals you got? What kind of progenies you got? Pink. It is intermediate between those two parents. And in F2 generation, what kind of phenotypic and genotypic ratio you got? 1:2:1. Both the ratios are same. So this is about the incomplete dominance. So when you analyze carefully, you'll be knowing that red and white are expressed under homozygous condition. They have got separate genes for their control, whereas pink color is expressed under heterozygous condition. Pink color is expressed under heterozygous condition. It is not having specific genes for its regulation. So these 2 points you have to write at the end. What you are going to write? So let me write those. Now just let me say you the conclusion of incomplete dominance. You have seen that the characters, flower colors like red and white, they both are expressed under homozygous condition. They are expressed under homozygous condition, and for their regulation they have got specific genes, that is capital R, small r, they are there. And pink is expressed under heterozygous condition. Pink is expressed under heterozygous condition, that is capital R, small r, and there is no specific gene for the regulation of pink flower color. So the intermediate color which is expressed is not having any genotype, any specific genes for its regulation. This we have to keep in mind. Whenever heterozygous condition will come, the pink color will get expressed. Now let us see the examples in the animal. One example I have taken, that is Andalusian fowl. Andalusian fowl. So here the 2 feather colors you will see, that is black and white. So black, according to the Mendel, it is, if it we consider this as dominant, we have to indicate that with the capital B and capital B, and the white we have to indicate with the small b and the small b. When we cross these 2, in the F1 generation, all blue colored fowls we will get. All blue colored fowls we will get, and when we cross F1 among themselves, self-breeding when we do to get the F2 generation, there you will get 25% black, 25% white and 50% blue colored fowls we will get. So in the market it is surveyed that the black and white color fowls meat is having less rate and the blue is having more. Fine. So that is you do this cross by yourself and just check yourself whether you'll be able to do it or not. So what did I told today? The important one. I told you the test cross ratios in monohybrid and dihybrid. Under what condition, what kind of ratios you are going to get. Like if it is heterozygous, 1:1 in the monohybrid, and in the dihybrid you'll get 1:1:1:1. And in the monohybrid, if it is homozygous dominant, you'll get 4:0. And there in the dihybrid, you'll get 16:0. And under homozygous recessive, in the monohybrid, you'll get 0:4. And in the dihybrid recessive, you'll get 0:16. that I told, and then we have discussed about the incomplete dominance in the snapdragon flower, which is important for your theory exam for 5 marks, as well as questions will be asked in the NEET and CET. And what is the important conclusion over there? So here in the heterozygous condition you will get an intermediate color, and for that color there are no specific genes. And one animal example we have taken, Andalusian fowl, which you have to do it by yourself. So in the next class we will see the next next example we will take, where it will show a deviation from the Mendel's principle.